Okay, good morning everybody. Thank you to ProImmune and the Museum of Science here for setting up this wonderful meeting and also inviting me to speak at it. So I'm going to talk to you today about a method I've been developing in the lab during my PhD to do high-throughput T-cell antigen profiling by deep sequencing. And so just before I begin, as Jeremy mentioned, I'm coming to you from Vancouver, BC. There it is. And we're located at the BC Cancer Agency, which is right here. And I work for Dr. Rob Holt there. We're part of the Michael Smith Genome Sciences Center. Again, as Jeremy mentioned, I'm in the 6th year of my PhD. And in the lab there, we have a whole range of different projects ongoing, from immunogenomics, synthetic biology, metagenomics, and cancer genomes. Okay, so back to T-cell antigens. So T-cell antigens are an attractive area of study because unlike any other component of the immune system, it's T-cells that are responsible for recognizing intracellular targets. And they do so by recognizing short, contiguous peptide fragments that are derived from intracellular proteins, but are presented at the cell surface in the context of MHC class I molecules. So that's illustrated here. We can see a normal healthy cell expressing its normal complement of proteins, which in the course of normal proteolytic cell turnover are converted into peptides, a subset of which can be loaded onto nascent MHC class I molecules in the ER, which then get trafficked to the cell surface. So this is a process that occurs, as we know, in virtually every cell type in the body, and it serves to provide a snapshot to the extracellular environment of what's being presented inside that cell. And that we can have CD8 T cells from the peripheral repertoire come by and inspect these peptide MHC complexes. And in the case of a healthy cell like this that's expressing normal self-peptides, the T cell does nothing. Both cells go on their separate ways. But obviously in the case of an acute infection by a foreign pathogen, we can get the expression intracellularly of microbial proteins, which also get converted through proteolytic machinery into microbial peptides, some of which can get loaded onto MHC class I molecules and presented at the cell surface. Now in this case, CD8 T cells in the peripheral repertoire can come by, inspect these, recognize them as foreign, and then mount an immune response that ultimately lead in the clearance of those infected cells. Likewise, we can have single base pair changes in the host germline coding sequences of the intracellular proteins, and these can lead to altered peptide ligands, which again can be presented on MHC class I molecules, and importantly recognized as well as non-self peptides, leading to their clearance as well. So this is the mechanism by which, as was mentioned earlier, the body can mount its own natural anti-tumor response, and it's the basis for which neoepitope targeting T-cell immunotherapies have been developed in recent years. Oh, the cell gets cleared. All right. Okay, so T-cell antigens are clearly a pretty important part of host cell immunity. But historically, T-cell antigen discovery has been a difficult thing to do because the T-cell receptor peptide MHC complex interaction is a complex one and is a difficult one to interrogate. Here we see a crystal structure of all of this interaction, and we can see right away that there's a number of different components at play, the center of which is that short peptide epitope that I was talking about. And so this epitope can be one out of an immense number of possible short peptides. So the first challenge in doing T-cell antigen discovery is to figure out how to sensibly search this enormous peptide epitope space. Secondly, this peptide is, as I mentioned, presented in the context of MHC class I molecules, which this MHC1 molecule can be one of thousands of different variants encoded by thousands of different MHC alleles. And each of these variants have their own preferences for peptide epitope binding, and also their own preferences for binding individual T-cell receptor alpha-beta complexes. So the identity, in any antigen discovery effort, the identity of the MHC class I variants that are present on host cells needs to be taken into account as well. Moreover, this TCR peptide MHC interaction is it's a relatively low affinity, transient, promiscuous one that can be modulated by the presence or absence of different co-receptors present on the respective cell surfaces. And importantly, T-cell activation, functional T-cell activation, requires the integration of a number of different biophysical parameters. Of course, we hear about affinity, and we know about affinity between the peptide itself and its MHC binding partner, and also the affinity of this peptide MHC complex with the TCR complexes that are interrogating them. But the kinetic on-off rates of each of these interactions, the peptide MHC half-life at the cell surface, and other things like TCR allostery influences on downstream TCR signaling are all important biophysical parameters that play into the activation of the T cell itself. So this is a specific example that serves to illustrate that point. So here we have two peptide epitopes. They're both agonistic epitopes for the mouse model TCR2C. And they're both, as we can see here, they're pretty structurally similar. And indeed, when complexed with MHC molecules, we find that the peptide MHC complex has a pretty similar affinity to the 2C TCR, certainly in the same order of magnitude. But if we look at their potency, their ability to actually elicit response from the 2C TCR, they differ by several orders of magnitude. So the point here is that affinity is not the whole story, although it is often used as a proxy for T-cell activation. Okay, so in the face of all this complexity, how are we as a community doing at identifying and characterizing T-cell antigens, T-cell epitopes? And the short answer is not great, but to put in perspective what I mean by that, we can go to the IADB, which contains the majority of known peptide epitopes that have ever been reported in the literature, and we can run a search for T-cell assays of any MHC restrictions from human and find that we get about 80,000 hits. So important to note here that these hits are unlinked. In most cases, the identity of the T-cell receptor that actually recognize these peptide epitope complexes aren't known. But so we do have 80,000 epitopes here that elicit reactivity in some context. But if we look at the flip side of the immune synapse, we see that in T-cell receptor profiling, TCR-seq studies are being done routinely. Thousands, hundreds, or if not thousands of TCR-seq studies have been performed, and in each of these studies, hundreds of thousands to millions of different unique TCR clonotypes are being discovered. So we can see here that T-cell antigen profiling has not kept pace with T-cell receptor profiling. And we also see that B, there's a huge wealth of T-cell receptor data out there that is, as we speak, being mined to find so-called interesting clonotypes. And they're interesting for various reasons, but problem is we really don't have any way of figuring out what those clonotypes are recognizing at a large scale. So what we need is high-throughput methods to be able to take large sets of possible peptide epitopes and then test them against these clonotypes of interest in order to begin to map the reactivity between T-cell receptors and this enormous peptide epitope space that could exist. Okay, unfortunately, existing methods for T-cell antigen discovery are not particularly well suited for high-throughput screening. If we have a look at some of these cell-based or some of these current existing methods, we see that we have cell-based methods that either require you to develop a reporter readout in the T-cell itself. So this is the case in cytokine release assays like ELISpot or NFAT-linked reporter assays. Or they require you to completely destroy the antigen presenting cell as a functional readout of T-cell activation. So this is the case in chromium release assays. So in situations where you have a T-cell clonotype or clone of interest and you would like to test it against a large set of candidate peptide antigens, these configurations require you to do so one by one, which testing each individual antigen in a separate well of a plate, for example. And obviously that becomes quite difficult to scale into large numbers. As an alternative, affinity-based methods have been developed where you can take single-chain peptide MHC constructs, build libraries of those, and deliver them into antigen display cells, which are generally yeast or insect display cells, and then fish for relevant antigen using soluble TCR reagents that are composed of your TCR clonotype of interest. So while these methods are scalable to high throughput, the drawback here is that they ignore physiological context. The natural antigen processing and presentation pathway is completely absent, and T-cell activation is not measured. These methods rely solely on affinity as an indicator of T-cell activation, and as I alluded to earlier, you run the risk then of discovering false positive hits or ignoring potential low affinity hits that are actually high potency. So I've developed a novel approach that marries the best of both worlds. It's a high throughput approach, but it also maintains that functional physiological cell-to-cell context. And it works by leveraging the exquisite specificity of the granzyme perforin pathway. So in vivo, when a T cell recognizes its target cell, it's able to deliver a class of proteases called granzymes, predominantly granzyme-B, into the target cell without allowing any to enter nearby bystander cells. And so this presents us with the opportunity to encode a reporter system in the target cell instead of in the T cell, like in the methods that I described before. And so I constructed this by taking a CFP domain, fusing it to a YFP domain, and in between those two domains, encoding a short granzyme-B cleavable linker substrate. This fusion transgene then is also encoded alongside short peptide-coding Minigene sequences that can be delivered together as part of this lentiviral cassette into surrogate host target cells. And once inside those surrogate cells, we get independent expression of each component. You see the short peptide coding sequence here, and then also the intact reporter. And if we look at cells that are expressing this intact reporter protein, we can interrogate them with a violet laser, and we see that instead of getting CFP signal that you might expect off the CFP part of the molecule, we get FRET transfer of that energy over to the YFP moiety, and we get yellow light off of this reporter molecule. And we can monitor this in FACS. If we plot FRET versus CFP signal, we get a resting FRET signature in the transduced target cells. So then, should there be a minimal epitope embedded in this Minigene sequence, it can be processed and presented and potentially recognized by our T cell of interest, at which point granzyme-B is shuttled over to the target cell. We get cleavage of the reporter molecule, and subsequently a loss of FRET signal coupled to a rescue of the free CFP signal. And that looks like this in FACS. So we get this pronounced shift as the FRET is decreased and the CFP is increased in these targeted cells. And this allows us to very readily gate on only the cells that are being actively targeted by our T cell population of interest. So this fits into our overall library screening scheme as such. So we can now then take large libraries of millions and millions of different mini-genes, encode them into a surrogate cell population such that each individual cell expresses a different mini-gene, and then co-culture this target cell library against our T cell population of interest. Perform the FRET shift assay, as I described on the previous slide, to isolate out cells that are carrying putatively antigenic mini-genes, and then recover the mini-genes that are encoded within them by PCR so that we can assess them by deep amplicon sequencing. So that's the principle. To do the initial proof of principles, I didn't start with large libraries. Rather, I just started with two model mini-genes. The first of which is derived from the chicken ovalbumin protein. It's just a fragment of that whole protein, but it contains at the center of it this synfecal minimal epitope, which is known to elicit reactivity from the OT1 model mouse TCR. And in parallel, I constructed the same mini-gene, the same fragment of chicken ovalbumin, but with the synfecal region scrambled. So this is my scrambled control. Each of those mini-genes were used to generate lentiviral reagents, which were then used to transduce a surrogate host target cell line, And then either of these cell lines could be cultured with or without expanded OT1 cytotoxic T cells. And so what this looked like is when you have either of the cell lines in the absence of OT1 T cells, we get this resting fret signature like I was talking about a couple of slides ago. And then upon introducing OT1 T cells to these populations, we get, again, this pronounced fret shift in cells carrying ova minigene, but then maintenance of that resting fret signature in the cells that are just carrying the scrambled negative control. So this showed us that the fret shift system actually works. The fret reporter is able to efficiently detect T cell reactivity. Well, the next question we had, though, is that exquisite sensitivity of the granzyme perforin pathway maintained in our in vitro screening system? In other words, is this specific? So to test this, I simply mixed together the ova minigene expressing cells and the scrambled control expressing cells together at a 1-to-1 ratio, and then screen that mixed population against OT1 T cells. And then, again, ran them through the fret shift assay and actually collected both these fret shifted cells here in this so-called targeted gate, and then collected a sample of the untouched population here that were not co-cultured with CTLs. I assessed either of those populations with qPCR using a custom assay that could distinguish between the ova minigene and the scrambled version. And we can see that in the untouched gate, all the minigenes that were detected were found at a roughly 1-to-1 ratio, as expected, because they were mixed together at that 1-to-1 ratio. But we can see in the target gate, all of the minigenes that were found, over 95% of them were of the correct ova minigene. So these data indicate to us that the reporter system is highly specific, that granzyme-B perforin specificity is maintained in vitro co-culture. Oops. Okay, so now it was at this point that I started going into library screening, and I did so by generating libraries of random minigene, starting from completely degenerate oligo that I then cloned into my lentiviral transfer vector and generated random minigene lentiviral library from. These random minigene viruses were transduced into the same host cell line that I used to make the ova minigene cell line to generate my random minigene expressing cell line. And then into that cell line, I spiked in the ova minigene cells at abundances starting from 1 in 10 all the way down to 1 in 10,000. And then screening each of those mixed populations against the spiked, or each of those spiked populations against expanded OT1 T cells. So the objective here was to see how low of an abundance of known canonical antigen could we go and still be able to detect it. So this is a depiction of that workflow. So each of the mixed populations were screened, as I said, against OT1 T cells. The same gates were collected as in the qPCR experiment. So I collected the targeted gate and I collected the untouched gate there. But instead of qPCR, I just did standard PCR and actually just isolated out the minigenes and submitted them for deep sequencing. So we got millions of raw reads from that, which were then processed and analyzed simply just by counting up the number of instances of synfecal that were detected in each gate of each spiked population. So those data are shown here. We can start at the left side of the slide, and we see that at the highest abundance, 1 in 10 ova minigene expressing cells, we see that most of the cells that are found in the targeted gate were of ova. And then we also see substantial enrichment of the ova minigene in the targeted gate relative to its counterpart untouched gate. And this trend continues all the way down to about 1 in 10,000 ova. And then if we shift over to the right side of the slide, we can zoom in at the 1 in 10,000 condition and look at every individual sequence that was found in that library. And so that's shown here. Each dot here is a sequence, and it's expressed, or it's shown as a relative abundance of that sequence in the targeted gate minus the relative abundance of that sequence in the untouched gate. We can see most of the sequences fall along 0 because most sequences were not enriched due to T cell pressure. But we see the red dot here is ova minigene. We can see that it is substantially enriched relative to background, over 10 standard deviations above background. And so these data indicate to us that this approach is able to match the sensitivity of the existing gold standard methods like ELISpot and so on, but with the added advantage of being able to simultaneously query over 1 million different unique peptide coding sequences. Okay, so encouraged by successful proof of concept experiments in mouse systems, I've started working now on adapting this whole system into human cells. And unsurprisingly, that presents a couple of new unique challenges, namely in how to obtain large numbers of appropriately MHC-matched APCs, and also large numbers of functional T-cell clones that we can use in high-throughput screening approaches. So I've started on a few different arms of approaches to try and address these challenges. The first and perhaps most obvious is to use our expanded CTL clones from a patient or a donor, and then screen them against autologous APCs that have been transduced with the Minigene-reporter viruses of interest. And so far I've been working with autologous B cells to do this. However, it's not always possible to get large enough numbers of autologous APC to do these high-throughput experiments. So as a second alternative, I've been testing out CTL clones against artificial APCs that are derived from the K562 cell line and have been lentivirally provided with the relevant HLA allele coding sequence. As a third alternative, in the event that you can't obtain a T-cell clone or expand it, again, to large enough numbers, but you do have an interesting TCR clonotype that you want to try and study, I've started also working on developing an artificial CTL chassis, which consists of the YT-Indy cell line. It's a natural killer-like immortalized cell line that has, again, been exogenously provided with the TCR sequence. In this case, we would have now a completely artificial screening platform that we could presumably be able to screen or expand out to hundreds of millions of cells and do very large high-throughput T-cell antigen screening. Just to provide you some preliminary data about that last one, just to show you that it's not all completely speculative, we've done some work to show that the YT-Indy cell line is actually a good artificial CTL cell line. We can see here right away that it has no natural cytotoxicity against the K562 cell line, which is good because that's our vehicle for artificial APC. But then we also see when you put in a CD19 CAR into the chimeric antigen receptor into the YT-Indy cell line, and then co-culture that YT-Indy CAR cell line against a CD19 expressing cell line, that we do get cytotoxicity in an antigen-specific way. So this tells us that the minimal signaling domains that are present on a CD19 CAR are sufficient to actually hook in to the signaling pathways of the YT-Indy cell line and result in antigen-specific degranulation. So now, as I mentioned, I'll be testing out a similar type of approach, but instead of a CD19 CAR, trying to actually reconstitute the whole TCR-CD3 complex. Okay, so I'll just finish off by talking a little bit about where we would like to try and apply this methodology to in a sort of medically relevant way. We think that this high-throughput TCR antigen discovery can aid in developing and improving biotherapies. And one way we think we can do that is by finding tumor target antigens. So it was discussed earlier today, and it's become a common sort of workflow to do whole exome sequencing and try and, from whole exomes, narrow down a set of mutation-containing neoepitopes that we think would elicit T-cell reactivity. But we would like to try and, with the scale afforded to us by this new approach, actually just generate a fragment library derived from whole tumor exomes and directly screen them and isolate out the actual peptide epitope coding sequences from the entire exome, rather than doing biased candidate selection type approaches. Secondly, we'd also like to leverage the high throughput ability of this assay to assess cross-reactivity. So T-cell cross-reactivity is a thing that everybody acknowledges must exist. We realize that a single TCR must be able to recognize a huge number of possible peptides, because if that wasn't true, if a TCR could only recognize one peptide, then the number of T cells that you would need to be able to provide adequate protection against the whole universe of possible peptide epitopes would require an organism that would be huge. It would, be twice the size of the CN Tower, for example. And so we want to leverage the high throughput capability of this assay to try and see if we can get a look at what this T-cell cross-reactivity looks like, and possibly try and find ways to predict whether potential therapeutic TCRs are highly promiscuous or are safe. And so this would have some real-world implications. So these are papers, complementary papers that were published from case studies that were done a few years ago, in which TCRs were affinity enhanced. They were engineered to have higher affinity for the MAGE-A3 epitope, which is a cancer-associated self-antigen. So this engineered TCR was then transduced into autologous PBMC and reinfused into the patients. And it was found in both patients that these TCRs were targeting cardiomyocytes in both patients. So this led to severe autoimmunity that was ultimately fatal. And this was because an epitope from the titin protein, which is expressed at high levels in cardiomyocytes, also elicited reactivity from these engineered T cells. So in this case, the investigators had no way of predicting that this would happen. There's no such assay that you can do as a QC metric to test if your therapeutic TCR will be safe or will have cross-reactive epitopes elsewhere in the exome. So this is somewhere we would like to try and explore somewhere down the road as well. Okay, so I'm going to stop there and acknowledge all of my lab mates, my home institutions, and also all the funding agencies that have supported this work. And I'd also invite everybody to come check out our lab website for updates on this and other projects that we have on the go too. And I'll also take questions. Thank you.
Thank you very much, Govinda. Okay, any questions? I guess. Oh, yeah. Right. Go ahead, Chris. No, you go.
Nice talk. I thought it was a really interesting screening idea. Can you comment on sort of how you optimize the time scale of the assay to avoid just granzyme-B killing the cells? And do you see any of that? And are you maybe missing out on really potent CTLs by avoiding dead cells anyway? Just curious about the actual technical aspects.
Yeah, that's a good question. And so I didn't show that data, but I've done that extensively. It's a lot of time course experiments, and we do find that there is what we call a safe sorting window, where you actually do get a period of about 2 to 4 hours where you can see, you can get up to peak fret shift signal and then have a period of 2 to 4 hours before you actually see cells start to drop out of culture. So we find that we don't probably lose very many cells at all due to degradation. We're able to capture them with this granzyme readout before those downstream effects occur.
Great. Okay. Thanks.
Yeah. I mean, I would imagine that would vary depending on what host cell line you use, but in the particular ones that we've used for these proof of concept experiments, that it was pretty robust time window.
Great.
You mentioned about the three different of approaches to the human screening. So are you now pursuing that third option of the artificial system? Is that kind of your primary?
I would say that's the primary focus at the moment. I've done some work on those first two screens, or those first two approaches, and we find with, like I said, with the autologous B cells, which is the cell type that we were working with, CD40L stimmed in vitro, that it's really hard to get them up to numbers in the hundreds of millions, which is probably what you want to be able to really adequately screen, more than 1 million different sequences, and moreover, they're actually quite difficult to transduce. So, and we sort of figured that was probably the best case cell type, and even that would be hard to scale to those levels. So, I think my personal view is that the totally artificial platform would be the best, but I think it would also require a lot of comparing to primary T cells in terms of their reactivity profiles.
Yeah, it's more a comment than a question. I mean, it's interesting that you mentioned I believe it was Immunocore study, right, about these cross-reactivities, and it didn't hit the news as much as TGN-1412, although the outcome was much worse. I mean, those two people, I think, died within hours of receiving the therapy, so there was nothing they could do, and it's a shocking result in a phase 1 trial, really. And I guess it just highlights the need for the understanding the immunosafety of some of these, potential vaccine approaches. If you don't know what if they're based on those sorts of exome approaches, you don't, it's hard to know what those cross-reactivities might be. And so certainly the need for understanding that better is absolutely there.
I have another question just in relation to I think one of your final slides about exome sequencing, obviously targeting that for individual cancer patients. What's the sort of time scale involved? The exome sequencing is fast, but then just to try and understand a bit more about your Minigene construct, obviously you just need to take that sequence, then generate the DNA, put it into a construct. So again, it's a matter of days to do that before putting it into your system. Is that?
Yeah, generating a Minigene library to be screened is very analogous to generating a fragment library to be sequenced. So rather than putting them on a sequencer, I would put them into a viral vector and then transduce and screen. So yeah, I anticipate, and just in practice so far, it is a relatively short timeline, weeks before we can get sequences of epitopes or putatively antigenic mini-genes so that we can then mine and verify and so on.
Of the 80,000 epitopes in the database, how many of those show up in screens on tumors? Like if you just look for exosomes from tumors, and what are we missing? It feels like normal epitopes maybe aren't as useful as mutated ones or otherwise altered epitopes.
Yeah, so that's a caveat that I didn't mention, that the IEDB deliberately does not include mutational neoepitopes. The only cancer-associated epitopes they would have are the self-epitopes, like cancer testes and so on. So in the cancer context, you probably wouldn't find any useful T-cell epitopes in IEDB. Having said that, I'm not really aware of any repositories where there are tumor neoantigens. So that would be a really useful thing to have. Yeah, I think most of them are individual papers where exome sequencing and epitope prediction have been done, but there's no central source, I think, for that.
Yeah, no, I think you have a really nice idea, and I'm just, I'm thinking about people like Rosenberg's lab, where it just strikes me that they must have a catalog of all the things they've identified over the years. It seems like it ought to be publicly searchable.
Yeah, that would, I think, be a huge resource. As you can imagine, though, and it was mentioned in talks previously too, that this would be a highly personalized approach too, just because the specific mutations that would give rise to targetable epitopes would be very variable from person to person. But as time goes on and more of these approaches are done, you could imagine also finding some overlapping ones that could be applied to other people, other patients.
Thank you very much.
Okay, thank you.